Hi there everyone, welcome to Saurabh's classes. This is Ashmita. In today's class, we are going to solve a bunch of questions from the IIT JAM Geology paper of 2023. Let's start with section B in this video. We are going to focus on 5 to 6 questions as I have told, and here this section will consist of 2 marks for each of your answers. Obviously correct answers. Starting with question 31, which of the following statements are about telophase? Okay, so over here, let us get to see the options first. Daughter chromosomes are yet to form. Option B says new nuclear envelope starts to reassemble. Yeah, here option B will go. This is correct about telophase. Division of cytoplasm begins, no. Nuclear membrane disappears, no. Now let us discuss in details about this phase of telophase, okay? Um, Telophase can be termed as the final stage of mitosis, okay? As the final stage of mitosis, where the separated sister chromatids reach the opposite poles of the cell and they start forming two nuclei. During telophase, there are a series of events that happen. We are going to discuss about those series right now. Firstly, new nuclear envelope starts to resemble rather around the separated sister chromatids, marking the beginning of nuclear formation. Okay, that is what I have marked in option B. This is what I was trying to say about telophase. There are other events that happen over here. We can talk about the other events like chromosomes. Talking about chromosomes, chromosomes begin to unwind and decondense into chromatin, which is the less condensed form of DNA. Okay. We can also talk about the division of cytoplasm over here, which begins by a process called the cytokinesis. Cytokinesis. Okay. All right. Over here, uh, two options are related to telophase. So let's go with option B as well as option C. I did not notice it first. Option B and option C both are going with telophase. So go with B and C. Both B and C are your correct options. So division of cytoplasm also begins by a process called cytokinesis, which I was talking about right now. Cytokinesis. Um, In animal cells, what happens? A contractile ring made of actin and myosin filaments forms around the cell's equator. Okay. Equator means the middle path and gradually contracts to divide the cytoplasm into two daughter cells. And in plant cells, what happens? A new cell plate forms in the middle of the cell. In animal cells, it happens around the equator of the cell. In plant cells, it happens in the middle of the cell, which will later in plant cells, a cell plate is formed in the middle of the cell, and this will later develop into a cell wall that divides the cytoplasm into two daughter cells. Now let's talk about option C a little bit in more detailed manner. Now division of cytoplasm begins, oh yes, we have already discussed option C, right, about cytokinesis. Um, let's talk about uh, the fourth option, yes. Fourth option, that is the nuclear membrane, it disappears in prophase, not in telophase. Okay, so this is related to prophase, and daughter chromosomes are not present during telophase. So obviously this is wrong. Daughter chromosomes are not there in telophase, and since they have already been separated in the previous stage of mitosis, which is called the anaphase. Before telophase comes this phase of anaphase. So over here only they have been separated. Okay. All right, so we have got B and C as our correct options. Let's move over to the next question. The characteristic morphological changes in cells undergoing apoptosis. Apoptosis are. Let's go through the option. Formation of plebs on cell surface, no. Uh, Swelling and bursting of cells, no. Collapse of the cytoskeleton, no. Condensation and fragmentation of nuclear chromatin, yes. This will go. What is apoptosis, first of all? Apoptosis. Apoptosis is a programmed cell death process. Programmed cell death process. And this happens naturally during development and tissue homeostasis. And there are several morphological changes that occur in cells undergoing this apoptosis process. What changes take place? Let us discuss now in details. There is cell shrinkage. Now, the cell undergoes a progressive reduction in size, and hence it is called cell shrinkage. What else? What other changes can we see? We can get to see formation of apoptotic bodies. Okay, formation of apop apoptotic bodies. This is another event that happens in apoptosis. Over here, during the formation of apoptotic bodies, the cytoplasmic contents of the cell are packaged into small membrane-bound vesicles, which is actually called the apoptotic bodies. Okay, and which are later phagocytosed by the other cells. Okay. Next, we are going to talk about our correct option, that is condensation and fragmentation of nuclear chromatin. Now, the chromatin over here, which contains the DNA and all the histone proteins, this chromatin undergoes condensation, okay, into discrete masses, which eventually fragment into smaller, denser clusters. All right. Now let's talk about what else takes place during this apoptosis program or process. There is a preservation going on of plasma membrane integrity. Okay, like unlike necrosis, what happens in necrosis? It's a pathological form of cell death which is associated with inflammation and damage to the surrounding tissues. At apoptosis is not like that. In apoptosis, what happens? Apoptosis maintains the integrity of the plasma membrane. So the formation of blebs on the cell surface, which is mentioned in option A, can occur during apoptosis, but it is not a defining feature of the process. So that is why I am not including this one under characteristic of apoptosis. Uh, Swelling and bursting of cells. Let's discuss the second point. Swelling and the bursting of cells is not a characteristic feature of apoptosis. Okay, this type of cell death, whenever it happens due to swelling and bursting of cells, this is called necrosis. Okay, which occurs in response to injury or infection, and it can lead to inflammation and tissue damage, due to which it can lead to the death of cells. Lastly, the C option, that is collapse of the cytoskeleton. This is also not a defining feature of apoptosis. Although some changes in cytoskeletal organization may occur during the process. Okay, some changes. So anyway, we have got option D as our the only correct answer. Next question you have is, a species of fish living in a lake are separated by drying up of the lake into two separate lakes. After several hundreds of years of separation, the two groups are unable to mate. These groups are now considered to be different species, of course. The others are not at all going. So species, different species of fish. Okay. Let's move over to the next question. Which of the following options represent the evolutionary relationship between the bird and bat wings as structures for flying? It would be homologous. Okay, that's your correct answer, homologous. Next question. Hyperventilation or breathing rapidly and deeply causes which of the following events in the arterial blood? Now, this will lead to increase in pH. Okay, so option C is your correct answer. Let's discuss the two fill in the blanks that is there. These will be the last question for this series. The net number of molecules of NADH formed from one molecule of glucose in glycolysis under aerobic conditions is 2. Okay, your answer would be 2. Next question. The number of possible unique combinations of linear tetrapeptides that can be made from 4 different amino acids using each amino acid only once in the chain are. Now this needs to be discussed in a little detailed manner. So now the possible number of unique combinations of linear tetrapeptides that can be made from 4 different amino acids. Here they are talking about 4 different amino acids. Um, Using each amino acid only once in the chain can be calculated as the first amino acid can be any of the 4. The second can be any of the remaining 3, of course. And the third can be any of the remaining 2. And the fourth can be the remaining one which is left. So we can say the number of possible combination is 4, then the remaining 3, then 2, then 1, and it would be 24. So there are almost 24 combinations, possible unique combinations of linear tetrapeptides that can be made from 4 different amino acids using each amino acid only once in the chain. Okay, so your answer would be 24. So that was the discussion for today's class. Hope so you all understood. If you have any kind of problems, you can ask in the comment box below. If you want to join our classes for live sessions, recorded lectures, and doubt clearing sessions, mock tests, and in any kind of interactive discussions, then you can definitely join our classes by dialing the number that is given in the description box, or you can also visit www.saurabhsirclasses.com. That's our official website. You can go to the description box below the video. You will get all the details over there. Thank you for watching and all the best for your exams.